I'm Wengong. I'm an assistant professor at Northeastern, and I have been at the Schmidt Center for 3 years as a postdoc. It's my great pleasure to introduce Hilary Finucane. She's an assistant professor at Harvard Medical School and associate member at the Broad, and assistant in investigation in the Translational Genetic Unit at MGH. Today, she will talk about her work, exciting work on variant scoring, and we are all eager to hear what she has done in the space. So with that, without further ado, please take it away.
Thanks very much for that introduction. I'm really excited to be talking to you guys today. Thanks very much for the invitation to speak at this symposium. So I'm going to start with genome-wide association studies. As many folks in this audience will be familiar with, genome-wide association studies have now identified tens of thousands of regions of the genome or genetic loci that are associated to common diseases and complex traits. So here's an example of a now old GWAS for schizophrenia, where you can see genomic coordinates on the x-axis. Each point is a variant. The significance of association is on the y-axis. And by analyzing genotyping data from tens of thousands of cases and tens of thousands of controls, researchers were able to identify over 100 genetic loci at which genetic differences lead causally to differences in disease risk. Each one of these green diamonds represents such a locus. And I just want to pause there, even though this is not news at this point, but just to re-emphasize the fact that we're able to make statements about causal human biology that leads to disease is really exciting. But of course, the big problem that the field faces now is we have all of these genetic loci, but how do they lead to increased disease risk? This is an enormous challenge for this field. We have these really exciting, promising little windows, but for each one, how are we going to figure out the actual mechanisms by which these genetic differences lead to differences in disease risk? So today I'm going to talk about two pieces of this puzzle. One piece is how can we take a region of the genome and refine it into the specific set of variants that are causally driving this difference in disease risk? And then what's a route towards starting from these variants that we have and trying to push forward towards the next step of understanding what they do. So to start by talking about identifying the specific variants, next slide. Uh-oh. Is this, can someone in AV help me advance the slide? There we go. Oh, whoop, 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 whoop. Back a couple now. There we go. Okay. So my group a couple years ago performed fine mapping in 3 large-scale biobanks. Before I describe the results, I just want to take one moment, since this is an audience with a lot of machine learning folks, to talk about the methodological piece of this. So I'm not going to say any details, but I'll just imagine that you have to do variable selection. You have tens of thousands of features. The features are highly correlated with each other. You have hundreds of thousands of samples. And the kicker is we really care about accurate quantification of uncertainty. You can't do lasso. MCMC is way too slow for this size data. Variational inference is also not accurate enough. So there's been a lot of really creative work figuring out how to do approximate these posteriors in ways that's accurate enough to be useful for what we're trying to do, but fast enough that we can then apply it to our 108 loci that we've identified in our GWAS. So, and my work has done methods development work in this space. My group has done methods development work in this space. A lot of what we've done has focused on robustness to model misspecification and how we can make sure that the results that we get at the end of the day are as reliable as possible. So we did fine mapping, next slide, yes, in 3 biobanks. So each of these biobanks has over 100,000 individuals. We're looking at tens of different phenotypes. By doing associations on all of these phenotypes at this scale, there's 16,000 loci to fine map. And this was approximately order of magnitude 1 million compute hours to narrow each of these loci down into the list of the most likely causal variants. This was led, oh, thank you. Excellent, thanks. So this was led by PhD students Masa Kanai and Jacob Eulersch and co-supervised by Mark Daly, and it was an enormous effort, which is described in more detail at that link. we identified thousands of likely causal variants, and while there's some caveats around the posterior probability of causality and how calibrated we think these numbers really are, this still represents a remarkable trove of variants that are likely to have causal impact on disease biology. To characterize these variants, we started by breaking the whole genome down up into 5 basic categories: exonic, UTR, promoter, enhancer, and other. And you can see genome-wide, most everything is other. But as you start to break down the categorizations of, next slide, this one doesn't seem to be working either, of these different variants, there we go. Then you can see that you see really large enrichments in these different categories, with the most highly confident variants really strongly enriched in the coding category especially. So now let's pause for a second and think about how cool it is that we've got fine mapped missense variants. So because of this really large scale computation that we did, and because of the methods development work that's been done in this field, we can now list 1,391 unique missense variants that are causally contributing to common diseases and complex traits. And so if we back up to our original question of, we've got all of these loci, how are we going to translate these loci into biological insights? Now we've taken a really important step, which is identified the individual variants. And for these missense variants, we can even say what the next step is. These are mutations that cause one amino acid to substitute for another amino acid at a particular position of a particular protein. And that is really, a big step forward from analyzing DNA mutations, but now we have to figure out what these amino acid substitutions do. And amino acid substitutions, of course, they can do a ton of things. On the lowest level, you've got a different set of atoms in different places at this point in the protein, and that means they're going to exert different forces on the other atoms that come near them. But that's only interesting because it means that the protein is going to be expressed in different quantities or different places, or it won't be able to adopt the right conformations in response to the right stimuli at the right times. So this is a hugely complex, multidimensional problem that we still are faced with. And this is where the AI comes in. So protein modeling, as Lucy talked about in the first talk, has made enormous strides. And this is a really exciting field. We now have seen a lot of leaps forward in prediction of a lot of different, for example, structure from sequence of proteins, a lot of these fundamental questions. So how about AI for understanding the effects of missense variants? Predicting the effects of missense variants is an extremely active field. So the Atlas of Variant Effects Alliance conveniently keeps a table where they're keeping track of all of the methods to predict the effects of variants, and it starts from classical methods like polyphen or SIFT, but if you scroll down, then you can see it goes all the way down to 94 methods as of pretty recently. There's an extraordinary amount of research in this space. And a lot of these methods are getting a lot of attention because they are solving a really important problem. And so this is a field in which there's been a ton of methods development. And the methods development has been extremely sophisticated also. It's reading these papers, you can see a very wide range of tools being brought to bear on this really important problem. So folks are using message passing neural nets on graph derived from protein structure. People are using extremely large language models trained on protein sequence databases, which I'm excited to hear more about in a future talk here. Variational autoencoders are being trained on multiple sequence alignments. The embeddings from the protein language models are being fed into population genetics models. The protein language models are being combined with the variational autoencoders and then calibrated using Gaussian process priors. AlphaFold fine-tuned on population genetics data with an auxiliary structure loss, and convolutional neural nets on voxelized protein structures supervised on population genetic data with auxiliary losses from both a variational autoencoder and a protein language model and a masked structure objective. Like, this field has just gone, has really seen a whole lot of sophisticated methods development. So with all of the brain power and computational resources that have been brought to bear on this problem, we were reading these papers and wondering, so which methods are best and how good are they? And that is a lot less clear than the fact that there's really cool methods development going on here. So just to give one kind of set of anecdotes from a series of papers, one task that you might hope a pathogenicity predictor would be good at is differentiating between de novo mutations found in cases versus de novo mutations found in controls for severe disease. And a lot of these papers are doing benchmarking on exactly this task, but they're doing it in very different ways and getting very different answers. So for example, in June 2023, we were seeing benchmarking on de novos using the Mann-Whitney U-test p-value. Shortly afterwards, different set of data, different metric. Now we're looking at AUROC. And now here, it's again de novos for severe disease, but again, the classification task is a little bit different, and we're using precision recall. Here, different data, different classification task. We're still talking about de novos for severe disease, but we're using a fourth evaluation metric. And all of these papers are coming out so quickly also that they're not, the methods aren't all being compared to each other. And so it's really difficult to kind of read through this material and understand where are we at as a field right now. So we came to this thinking it's time for some independent benchmarking using human genetics data, using the metrics that we know are most informative about the outcomes that we care about the most. And one big inspiration for this work is that in the deep mutational scanning field, there's been some really great efforts to systematize the benchmarking and then to have independent groups come and benchmark a whole bunch of these different methods. But for human genetics, things are still pretty scattered. So we wanted to ask three main questions with this work. First is just a metrics question. What should we be using as our metrics to evaluate these methods? And then also to expand past there's one dimension on which methods can be better or worse at, and instead say there's a wide range of, even within pathogenicity prediction, there's a wide range of possible pathogenicity tasks. So how are these methods performing at different parts of the selection spectrum? And also, how does performance differ for within gene tasks versus across gene tasks, which are two very different tasks? So Konrad Karczewski and Sophie Parsa and Jeremy Gez in his lab, together with Masa Kanai, whose name got cut off the slide, apologies Masa, together with several other collaborators. And I set out to do this independent benchmarking. And we are calling our framework Genetics Gym, inspired by Protein Gym from the Marx Lab. And we did four main types of benchmarking to begin with. So all the way on the heaviest selection end of things, we are returning to de novo variants for severe disease. And then now we're looking at rare variants for neuropsychiatric disease, rare and low frequency variants for common diseases and complex traits, and then common variants for common diseases and complex traits, which are these fine mapped variants that I described in the first part of the talk. And so here are a subset of our results that we found using this benchmarking on a subset of the data. And I think that there are a few main takeaways here. One of them is that the methods that are doing really well in the heavy selection regime are different than the methods that are doing really well in the lighter selection regime. And this isn't that surprising. These are very different tasks, and selection is doing very different things in these different places, and a lot of these methods are trained with a lot of evolutionary data. And so it makes sense that we would see really different tasks. But I hope that, or different performance, but I hope that what this does is it allows the field to start to move away from saying we have the best pathogenicity predictor to saying here's the particular area that we're focusing on, here's the area that we now are doing well at. And that we're going to be able to see papers that instead of saying we're the best at everything, say like this is specifically what we're good at and here's our relative performance at these other tasks. So when you're doing pathogenicity prediction and you're trying to differentiate a set of disease variants from a set of control variants, to identify those variants, there's two pieces, identifying the gene and then identifying the variant within the gene. And we wanted to tease apart those two different components. Because looking at this, it's really unclear to what extent, for example, our PopEve and misfit methods that are doing really well because they can find the gene versus they're doing really well because within a gene they're doing, they can identify the damaging mutations. And so what we did is for each one of the methods, we converted the method into a gene score just by taking the median score in the gene. So now we're going to say for each method, it's got its high-scoring genes and its low-scoring genes. And just on the basis of those gene scores, how well will they do, these methods do, on these exact same benchmarks. So here the red is the same data from the previous slide, but now we've broken it down so that the blue shows you the performance you would get with only the gene score from the same method. And to me, this is actually pretty surprising to see that the gene scoring is not a huge component of accuracy for de novo variants. Another thing to note is that the relative performance of the methods is really different. It's not the case that the methods that are doing really well at pathogenicity prediction overall are the same as the methods that are doing really well at gene scoring. In fact, you can see that MPC is the method that does the best at gene scoring across the board, but of course there's a need to go further and look at methods that were designed specifically for gene scoring and start to compare them and see both benchmark the performance, and then also I think that this shows that there's a lot of room for potential improvement in this space. But this figure still doesn't get at this really important question of which are the methods that are doing the best within gene. We have, for example, there are a lot of different applications. So you might care about distinguishing disease variants from control variants for diagnosis, but you also might be conducting a burden test using exome sequencing data, in which case what you care about is only within gene performance because you're never going to compare a variant from one gene to a variant from another gene. The tricky thing about benchmarking within gene performance is you need a lot of data. And so these data sets are too small for us to start to build case control comparisons within each gene, which is what we would like to do in order to get at this really important task. And so we developed a new data set using a large population genetics database, GNOMAD, but we used new data from GNOMAD, and we were careful to exclude the data that was used in the training of these different methods. And so by excluding any site at which variation had been observed in the time period during which these methods were trained, we were able to come up with an independent data set that then, because it's based on population genetics data, had enough variance that we could do within gene benchmarking. And so looking at this independent benchmarking using population genetics data, we could see that if you look again just at the overall performance, the same methods that were doing well in the heavy selection regime that are really picking out variants for de novo disease, for example, de novo variants for severe diseases, are also doing well at this task of differentiating between observed and unobserved variants in a population genetic database, which is what we would expect to see. But then if you take this same data and you now do within gene evaluations and you average over all genes, then all of a sudden the results really shift around a lot. And in particular, ESM, which in our genome-wide comparisons wasn't as competitive with the other methods, as a within gene method, it does extremely well. And I think that we're going to hear more about ESM shortly. So to conclude, different variant scoring methods have different levels of accuracy at different parts on the selection spectrum. I think this is a really important thing for us to keep in mind as we're following this super active field. And there's also a difference between differentiating among variants within a gene versus differentiating among variants across many genes. We've released our data publicly, and soon we'll be making our code publicly available as well. And we're hoping that that is going to make it really easy for future methods developers to benchmark in a way that will make it easy for us to follow what's going on and how excited we should be about which innovations. And then just to return to this initial question where I started, it's very clear now that AI is extremely powerful for characterizing missense variants, but this question of being able to differentiate between disease variants and control variants is a different question from the multidimensional characterization of what a variant does to protein structure or protein binding or protein stability. And so I think that a direction that it's really important to be focusing on now is away from the very well, established prediction problem of pathogenicity and towards the much more difficult and complex problem of characterizing that function in all of its multidimensional complexity. So I'd like to thank the collaborators on this project, and I'm happy to take any questions.
Well, thank you for the wonderful talk. Any questions? We have maybe 3 minutes just to keep us on time.
Thank you, wonderful talk. So can you comment, is it possible if we can build an ensemble of methods? You talk about various methods. We can build an ensemble method and a generative adversary network focusing on the misfitted data points, so we can improve the performance of current available methods.
Yeah, I'm super excited about ensembling these methods. I think the thing to think about is exactly what the task is that we're trying to do and what the data is with which we're going to train our ensemble. So for example, one thing I've been thinking a lot about is burden testing. And there, if you want to ensemble method, and specifically, how are we going to ensemble these methods for really powerful burden testing? But what's tricky there is the optimal ensemble is going to depend on the gene. And so I think that trying to build a framework in which we can understand in a focused way what we're trying to get out of the methods, and then figure out what kind of data we have to supervise an ensemble, be very careful about double dipping, then I think that there's huge potential.
Thank you.
Go ahead.
Question. So there is a very large gap between a variant and pathogenicity, encompassing like multiple length scales.
Yeah.
But so do you think that collecting data at these different length scales and sort of perhaps like bootstrapping all the way up to an organism level characterization, which is like, is this pathogenic or not? Is this something that you find useful?
Yeah, that's super interesting. So I think that if all we care about is pathogenicity, then an extremely useful type of information is evolutionary information. And you see just a huge gap between methods that are taking advantage of either cross-species or within human conservation or constraint versus methods that don't. And so I think that in some ways that if what we care about is pathogenicity prediction, then that means we can skip a whole lot of function and rely really heavily on evolution. I do think that the better we can understand function, or maybe what I'm saying is the only way to get there is not just to understand function. Evolution is giving us a huge amount of information as well. I think that the better we can understand function, the better we'll be able to do pathogenicity characterization, though there's a really high bar to be able to do something that's going to be informative on top of a lot of these evolutionary-based methods. But I think that pathogenicity prediction is not the only task out there. And so to me, the idea that you would be collecting a lot of data at multiple length scales and trying to characterize mutational effects at these different length scales is awesome in its own right. Like regardless of whether or not that's going to help us with pathogenicity prediction, we have this other task, which is super foundational. And so I think that I'd be excited about that for those reasons.
Thank you.
So maybe one last question, just for the sake of time.
Yeah, this is a quick question on kind of the separation between coding and non-coding variants, and if there's a lot of confounding between, conservation or other mechanisms that perhaps are in your next steps.
Wait, sorry, say that again. There's a difference between non-coding and coding variants.
Right. So, as just discussed, there's a conservation difference between them. Right?
Yeah.
But in terms of your next steps and structural regulation, that's where non-coding really shines.
Yeah. So I think that non-coding variants are just much harder than coding variants. And so I think that to me, there's kind of two different tracks of research. And one of them is let's take these coding variants because we already have such a big head start and let's run with them as fast as we can. And then at the same time, let's think about these foundation models for understanding non-coding variants, but where the AI is not seen as much success in that area. And even before we get to the AI methods, we don't even know how to connect these non-coding variants to their target genes,
Exactly.
in a lot of cases, which is a problem that I have thought about a great deal. We're making a lot of progress, but it's still something where it's not like having a missense variant where you can can just say like, oh, here's the first thing it does, what's the next thing it does. So yeah, I think we got to keep on working on that, and that's just a much harder problem.
Thanks.
All right, thank you. Let's give one more round of applause.
Thank you.
Okay, so our next speaker is Mehrtash Babadi. He's the director of the computational methods in data science platform at the Broad Institute, where he is also an institute scientist. He has done a lot of exciting work in the single cell space, and he's recently very excited about single cell foundation models. So today we will hear about his talk on CellariumGPT and CellariumCS. So with that, Mehrtash, please take it away.
Thank you very much. Is this the slide advancer? Cool. All right, so excited to be here. So today I'm gonna talk about two of the projects that we've done recently. But before that, I wanna start with a few slides of dogma. And I'm gonna be careful with not to spend all of my time talking about the dogma because I've done that in the past. All right, so since the advent of the single cell technology, we've been building these cell atlases, lots of data, hundreds of millions of cell state observations, across different conditions from different donors in health and disease. So the question is that what do we want to do with all this data? Why are we collecting all this data? How can we sort of put this data to good use, better use than what it was potentially intended for originally? So I would like to sort of go back to another project that generated a lot of data, and that's the Human Genome Project. That, there were like many sequence short reads. All these reads were aggregated, they were indexed, and then the final outcome of that was a FASTA file. One long file with like 3 billion entries. I'm definitely oversimplifying it, but now we are collecting lots of cell phenotypes. We have like trillions of cells in living organisms. We're not even like nowhere near there, but we're just collecting lots of data. So the question is that what is the final product? So I don't think that a final product of this is going to be a database or a file. It can't be that. The final product, in my opinion, initially is going to be a search engine, so that it can basically make all this data findable, and then a foundation model. And I have like very strong reasons for saying this. So the Human Genome Project, it was a point estimation problem. We wanted to basically sequence one genome, find basically one point in a huge space of all the possible sequences. But whereas understanding human cell, a eukaryotic cell, is all about basically understanding distributions or conditional distributions, like what cell state can we have in this context given this genome? Or if you're interested in dynamics, you want to know basically how does a cell state change in response to a certain action given a certain context and given a certain genome. So very different problems. One of them is to basically just estimate one point. The other one is basically just learn a dynamical system. A cell is a dynamical system. It's not a static thing. So, and I think the search engine itself is also going to be a band-aid, much like conventional search engines are band-aids now, and they're being replaced with AI-based search engines. So I think what we're left with is actually just a foundation model. So my dogma is that the final product of a successful human cell project has to be a foundation model. And I'm saying it not in a buzzwordy way, in actually a very real, actual, visceral way. We really need to do this. This is the way to do this. There's no other way of doing it. All right. So now take it to the practical. So I've been involved in a project at the Broad, led by Steve McCarroll and Evan Costco, the Human Brain Variation Project. This single project is going to produce anywhere near 200 million to 500 million multimodal single cell observations. Now we're thinking about even more ambitious projects like Project Radical that is going to look at dozens of cells, many variants across conditions, do multiple readouts, and probably it's going to produce anything from 1 billion to 10 billion cells. So I'm not an engineer, but I realized that we actually need to have engineering chops. So I basically put my engineering hat on about 3 years ago, and I basically started leading a team that built this product called the Cellarium AI Platform. So what Cellarium AI Platform is, it's a cloud-native sort of software infrastructure for data operations and machine learning operations that scales to billion-scale data sets and beyond. It allows sort of storing and querying lots of data, rapid model development, testing these models, and turning models into products, into sort of services that the community could use. The platform has like multiple things inside it. There's a data storage layer. There's a machine learning library that basically in which we implement how we can train large-scale models in distributed environments on lots of data. And there's some microservices that allow us to glue things together. And ultimately, everything culminates into building applications. OK, I'm not going to talk about the library itself. It's open source. It's documented. I encourage you to go read about it. But I'm just going to talk about applications. So the first application is what we call Cellarium CAS, or the Cell Annotation Service. And this is a work that is led by Fedor Grab, Muscles, and Kevin at the Data Sciences Platform. And it tries to address the first problem, sort of the Band-Aid problem, of building a search engine for all the cell data that we're generating. Why is it useful? Well, because we need to be able to contextualize cell experiments. We need to find, like, this specific cell state. Is it similar to something that I've seen before, or is it something novel? So that's sort of the problem that CAS solves. You might have used Google inverse image search that is now called Google Lens. You're basically doing the same thing for cells. So the idea is that when you have a new single cell data set, you're sort of embedding each of your cells into a vector space. We have already done that for hundreds of millions of reference cells. And then we're sort of doing a vector search in that space. So what is basically enabled doing this project is sort of learning like highly informative embeddings over large amount of reference cells. And now, once every cell is sort of endowed with a number of similarity hits in your reference data set, you can actually look at what metadata is associated to each of those cells and sort of summarize it and return it back to the user. That's sort of what we call annotation, which is a generalized form of cell type annotation, but cell type annotation is certainly one type of annotation that we can return. So there's a lot of engineering machinery under the hood to make this sort of a performance service that sort of scales to hundreds of thousands of requests. I'm not going to talk about that. One thing that I do want to say is that summarizing what the community have said in the past about a data set or a cell is actually a very difficult task because people use different terminologies to refer to different things. So it's very important to use ontologies and sort of use the relationships between the labels to be able to sort of summarize and build a consensus. And that's also something that is baked inside the cell annotation service. So the way it works is that the interface is that sort of you bring a fresh data set, and all of that is happening under the hood. And then what you get as a result is if you're doing cell type annotation, you get the cell type ontology with all the ontological terms that are relevant to this data set, with scores and relevance color-coded. And now you can select your favorite ontological terms and sort of highlight the relevance of that term for every cell in your data set. You can do this, you can do this. We released a preview of this back in July to a number of broad labs. A public beta of this will be released next week. And just yesterday, 10x Genomics, they basically released a beta version of this in their sort of cloud analysis software suite. So this is work that we've done in collaboration with 10x Genomics. If you want to try it for yourself, please just go to our website, cellarium.ai. There's a wait list that shortly you'll receive an invitation link to use the service. OK. So let's switch gears, talk about the second project, which is the CellariumGPT. This is the one that I'm personally more excited about. So the next frontier at the intersection of AI and cell biology is to be able to do in silico cell biology. And what I mean by that is to be able to, let's say, predict something that you haven't observed about a cell, such as one missing modality from another modality. Or more interestingly, to be able to answer questions such as how can you reprogram cells in response to certain interventions, chemical perturbation, a transcription factor overexpression, a CRISPR intervention. And the holy grail of that is that how can we remodel tissues? Like how can we take sort of a fatty liver and predict computationally what kind of intervention would actually remodel this tissue into like a healthy phenotype? A very daunting task. But this set of problems are very different from a search problem. This is context-specific prediction task and requires us to sort of learn the language of cells and tissues. Which is sort of the second thing that I incited my dogma that we should do about cells. All right, so we're using transformers to do this, and the reason is that transformers are just superbly useful for this task, for at least two properties among many other properties. One is that transformers are basically invariant to permutation of the input, and the facts that we know about the cells, these are not ordered. Like that this gene is expressed, that gene is expressed, you have this surface protein, the chromatin is in this organization. So these are like independent set of facts. So if you want to sort of learn sort of a joint distribution of all of these facts, transformers are really good for that. The second reason that transformers are useful is that in the same sense that the usage of language in natural language is very context specific and context dependent, in a sense, the words sort of get their meaning from the nearby words. The same thing can be said about the gene function and the function of gene products and the way basically gene products interact with each other to sort of modify the function of each gene. It's very much like how things happen in the natural language. And transformers are equipped with the right type of implicit bias, the right type of structures, such as self-attention, to actually be able to, to learn these things from data. So what we have done is that we've basically taken lots of single-cell transcriptomics data, and we have built a vocabulary for this data. And this vocabulary includes context of this experiment that this cell is coming from, and also the observed cell state, in this case just the transcriptomic cell state. So we still haven't really done anything in the space of really connecting this all the way back to genomics, and in my mind that's the next frontier. And then what happens is that after we build this vocabulary is that all of these tokens will be embedded, very much like how in natural language text pieces are being embedded, and then everything goes into a transformer stack, and then we make a bunch of predictions, such as predicting some of those context-dependent labels, and also like predicting the count distributions. So there's a lot of details that goes on there, like how you actually train these models efficiently, and what type of regularization you use to basically learn. And we're going to have a preprint on this soon. But let me just show you some results. So the very first result is basically just to check how much immunology your model knows about. Like, does it have like basic grasp of hematopoiesis? So what we've done is that we've just taken two cells, let's say like a naive CD8 positive T cell, and then an effector memory CD8 positive T cell, and then we looked at what are the genes that are like commonly expressed in the two cell types, and what are the genes that are only exclusively expressed or like more highly expressed in the effector cell state. So we prompted CellariumGPT basically with a series of prompts and ask a series of queries. So the prompt says that basically we start with just the common genes, add one at a time, and then we start adding those like effector genes. And then we're asking, for the query, we're asking again some genes that are in the effector state and some genes that are in the naive state. And here's the result. So like NKG7 and granzyme A, these are two genes that you expect them to be expressed in the effector state. And we can see that the moment we start prompting the model with cystatin 7, then the expression level of NKG7 goes up. And also the same thing happens for granzyme A, and it continues as we just prompt the model with more and more of the effector genes. Likewise, if you look at, for example, TCF7 and LEF1, that are genes that are like just generally lower expressed in the naive state, we see that as we basically tell the model that this is an actually effector-like state, the expression values of these things go down. The other very cool thing is that if you just look at these distributions, and these are distributions of copy numbers of mRNA, they very much look like mixtures of negative binomial distributions. But we never taught the model that. We trained the model in a fully non-parametric way. But the model has actually learned from this distribution of counts in the single-cell data sets that negative binomial distribution is actually a good distribution for describing the outcome of the experiments. This is super cool. All right, so the next thing we did is that we basically asked that, okay, so now that we have this model, how can we actually extract some biology out of that? So what we did is that we performed a series of experiments. We just take one cell, and we take one gene, and we just reduce the expression level of that gene, and leave every other gene the same. And we repeat this for every other gene. And we give these as prompts to the model, and then basically query what the model thinks the expression levels of every gene should be in the cell. So what we would expect to happen is that if there is some co-regulation or covariation sort of is going on between a group of genes, since every other one of those genes are left intact, they will sort of pull up the expression level of the gene that you sort of knocked down, but the gene that you knocked down will pull down the expression levels of the genes that are sort of covariant with it, right? And then so what we can do is that we can compare the output with the baseline and sort of calculate some log-fold change with respect to that baseline. And we can think of these outputs as sort of a covariation signature of every gene. And now we can build a gene network out of that based on the cosine similarity of these covariation. So let's just see some results. So we took one mighty cardiomyocyte that we have a lot of it in our training data, and we basically did that to it. And this is sort of what we get out. So in this plot, every point is a gene, and the points that are close to each other, these are basically genes that have similar covariation structure. Okay. So we clustered it, but now the question is, what do we do next? So Churchill has this very famous saying that gene set enrichment analysis is the worst form of single cell analysis, but it's the best. But it's, except for all the other forms that we've tried. So that's what we did. We basically took every one of these clusters, and we did gene set enrichment analysis to see whether this cluster corresponds to one of the known gene sets. And lo and behold, it does. So that tiny dot over there, if you zoom in, these are all of your mitochondrial genes. If you zoom in on the down below, these are all the ribosomal subunit genes. If you look like right there, these are sort of UPRs. If you look down below, this is the electron transport chain genes. And then if you look in the middle, you see the cell-cell junction organization genes. And just right somewhere in the middle, you see all the genes that are involved in muscle contraction and sarcomere organization that you expect to see in cardiomyocytes. And also myofibril assembly. So we basically see core functions. We also see cell type specific functions just popping out of this model that is trained on lots of observational data. So we tried something else. We also basically tried to give this model an H-tert immortalized RPE1 cell line. And this is something that is not in our training data set. This is just a new cell type, so far as the model is concerned. We repeat the same experiment, and this is the map that we find. Again, if you look at, do the same sort of clustering and enrichment analysis, we find again up there, these are all the ribosomal genes. Here we see cell cycle genes, all of them. This whole left island, these are cell cycle genes. And this is very important because these immortalized cell lines, these cycling cell lines. And so that's exactly what you expect to see, among other things. The other thing that is very important is that CellariumGPT had never seen cycling RPE1 cells. We only trained CellariumGPT on primary cells. And primary RPE cells, retinal pigment epithelial cells, these are all post-mitotic. They don't cycle. But we give as input to the model a cell that looks like something that was in the training data set, but it was cycling. And CellariumGPT understood it in a zero-shot setting. And this is, in my opinion, an instance of basically in-context learning. Just basically observing something new and sort of being able to synthesize facts about it from the rest of the data. So the other thing that we did is that we sort of tried to compare the output from CellariumGPT with actual CRISPR perturbations. But we have to be careful about that because what we see in CellariumGPT, these are basically covariation structures that in observational transcriptional states And so you can imagine that there's sort of homeostasis manifold for all the cell states. And then you have oxidative stress context, you have like hypoxic context, you have different donor genetics, cytokine stimulation, transcriptional fluctuations. And because of all these sources of variations, the gene expression sort of lives on some type of manifold. And that's sort of what CellariumGPT is picking up on. But an actual perturbation experiment is a bit different beast because you're basically acting on a cell. And then the cell can potentially even go off of the homeostasis manifold. So in a sense, it's sort of comparing apples to oranges. So we don't expect right now CellariumGPT to be able to predict CRISPR perturbation responses. But nevertheless, we tried that. So on the right, you see basically the same gene network from an actual CRISPR perturbation, and on the left, the same gene network from that I showed you before. We can sort of try to color them based on the Spearman correlation of the responses. So a blue gene is a gene that the covariation pattern that we see in CellariumGPT is compatible with the CRISPR response, and a red gene is a gene that is completely incompatible. And we're just starting to look at this more closely, but what we see is that cell cycle, DNA-dependent DNA replication, anything that is to do sort of with cell cycle, these are sort of the same between the two setups, and we believe it's because a CRISPR knockdown of a cell cycle gene arrests cell cycle and brings down every other cell cycle gene together with it. And that's sort of why we see compatibility. But if you look at some core cell functions, such as protein synthesis, mitochondrial translation elongation, nuclear export, the CRISPR response is very different from a covariation structure we see. And because if you just knock down one of the ribosomal subunits, the cell dies eventually, right? Okay, so quick conclusion and outlook. So Cellarium Cas makes about 100 million publicly available single-cell data inverse searchable, and totally encourage you to just go on our website and sign up for the service and try it out and give us feedback. CellariumGPT, I think it has achieved a remarkable grasp of cell type-specific cellular processes, and we will openly release all the code and all the model weights as soon as we're in a good shape to do that. More generally, I think AI really presents an unprecedented opportunity for cell biology research. Like, we have all the evidence for that now. The other piece of evidence that I guess we have is that top-down approaches are better than bottom-up approaches. This is something that we've seen in natural language. We didn't learn to synthesize text by starting from grammar and syntax. We just fed a lot of text to the models. We didn't sort of learn to design proteins by doing like quantum physics and simulations, even though we should do that probably. We basically just did lots of cryo-EM and trained models and just that scale. I think there's a good philosophical and physics-based reason for this, because every system has its own level of description at different scales. So cells has its own sort of emergent level of description that you can basically learn cell function by just looking at cells at that scale. You don't need to go and really understand mechanistically how every single protein interacts to be able to say something meaningful about cell states. Of course, if you want to be able to modify cell state, that's a different story. So that's why you need to ultimately connect all of this back to biochemistry. But I think we can make a lot of progress in understanding cells and understanding how cells respond to interventions just by looking at cells at sort of, at the highest large scale lens. The outlook is that we need to develop standardized quantitative benchmarks for all the cell language models, because that's how you can make progress. Then fine-tuning CellariumGPT on various prediction tasks, such as chemical and CRISPR perturbations, viability screens, and so on and so forth. And then perhaps calibrating the confidence of these types of cell language model predictions towards sort of making progress in the lab in the loop type scenarios, so that we can use these models to sort of guide generation of new data sets. And ultimately, I think the holy grail problem is how we can marry biochemistry and structural biology models with cell language models. That's one of the hardest problems. So with that, let me just get to my acknowledgments. So great thanks to CellariumGPT development team, Stephen Fleming and Yerdas Ordabayev. Specifically like Fedor Grab for Cellarium Cas, folks in the data sciences platform, Broad Legal, Eric Lander. So I've been meeting with Eric Lander. We have been meeting with Eric Lander for the past 2 years. And we've benefited so much from the guidance that we see from Eric Lander. I also lost half of my hair. I think it's I think it's worth it. I still have more hair, Eric, so. Steve McCarroll from the Center for Human-Brain Variation for supporting a lot of the works that you're doing on Agreca for super insightful discussions. 10x Genomics for supporting cell annotation service. CTI Cell by Gene project for making a lot of standardized data available to the community. And a lot of the interesting things that are happening right now is really just owing to them for like really standardizing all the data so we can actually do machine learning on that data, because otherwise we just couldn't. We had to spend all of our time curating the data. And lastly, folks from Cerebras who gave us access to their wafer scale GPU, well, wafer scale devices, which are some of the most marvelous pieces of chips that I've seen in my life. So we've been sort of training all of our models on Cerebras. And finally, like folks from the Argonne National Lab for allowing us to sort of use their computing facility. Thank you.
All right, thank you for the wonderful talk. Actually, we've been talking a lot about this space as well, and I should worry about my hair. We are running a bit short on schedule, but maybe one or two questions real quick so we can give Alex more time.
Hi, thank you very much for a very interesting talk. Just a couple of quick questions. So do you reckon this CellariumGPT can potentially be used, for example, for identifying covariant genes to develop probes for targeted spatial transcriptomics? And also, do you see an opportunity to study these covariation, to take these covariation embeddings and apply them in context of case control data sets to see what groups of genes are covariant between like disease and control?
Yeah, both of them are great questions. The answer to both of them is yes. So definitely, you can identify. You can basically start with a gene set, and you can use the covariation pattern to sort of extend it to a larger gene set. And perhaps, some of them are better targets depending on the assay that you're designing. So definitely that. And yes, so the responses that you get from CellariumGPT, these are like cell state specific. So a very intriguing question is to basically just see, what would be the difference between the gene network that you get from the healthy state, wild type state to sort of a disease phenotype. So we are in the process of doing these types of things, but I don't have anything concrete to share at this point.
Maybe let's give a chance to the folks in the back. They're harder to come up stage. We can definitely accommodate your question offline. Yeah. Let's see one hand in the back. Or hold on. Did I miss something? Sorry.
So, just
okay, go ahead.
Quick question. Can you use CellariumGPT to create biologically meaningful embeddings for single cells or cell lines?
Yeah, great question. So the problem of like embedding is a very loaded thing. So why do you want to embed something? Because if you want to embed something to be able to find similar stuff, in that embedding space. But what is similar stuff? So do the two cells, every cell has like, billions of like, well, not billions, but like thousands of different functions, right? So you might be actually interested in an embedding that emphasizes on like an inflammatory response pathway, right? Mm-hmm. So there is no single embedding that you can make. So I basically want to say that maybe that's not the best question to ask, even though we can get like a million different types of embeddings. So whether this embedding is better than somebody else's embedding, it really depends on what task you're subjecting these embeddings to. So perhaps maybe a great question, which I'm inspired just by your question, is to basically ask, like, can we use like stuff like CellariumGPT to build embeddings that sort of emphasize on like different aspects of cell biology? And perhaps we can. Yes.
I just thought the model structure was a bit similar to the universal cell embedding model from Jure Leskovich or the recent SC Foundation model that was in Nature. So I thought maybe it would be possible to use for cell presentations.
Yeah. So there are a number of similar efforts in the community. SCGPT, GeneFormer, SC Foundation, and again from each of these you can get embeddings. But again, if you don't guide these embeddings into the directions that you actually care about and for the downstream biology that you're doing, the quality of the embedding is just a toss-up. It's basically just based on the biases of the model that you have. So you can get embeddings and use them for different tasks, but I think I would start from the task and then go ask, like, how can I make an embedding that is useful for that task?
Thank you.
Okay. Let's give Mehrtash one more round of applause.
Okay, so it is now my real pleasure to introduce our second keynote speaker, Alex Rives, who is very soon going to join us here at the Broad as a core faculty and also the Department of Electrical Engineering and Computer Science at MIT. So we can't wait to actually have you here whole time. So it'll be really exciting. So Alex is an expert in developing AI models very broadly for scientific discovery, for scientific understanding, for scientific design in biology. And he is currently founder and chief scientist at Evolutionary Scale. And I hope many of us have seen in June the new model of ESM3 being released, and I'm sure you will be telling us much more about it. So welcome, Alex, and the stage is all yours.
All right, thank you very much. There we go. Okay. Well, I'm going to be talking about our work over a number of years now, so I guess about 5 years, developing language models for biology. And so through large-scale gene sequencing efforts, we've collected the sequences of billions of proteins across the tree of life. And if you think about these sequences, they're written in a language that is incomprehensible to the human intellect. And so kind of the question of ESM research program is, can we learn biology? Can we learn to understand these sequences through language modeling? And we have the technology to read and print genes. And so we have biology is fundamentally programmable at this very basic level, but in order to program it and to go beyond sort of taking the parts that biology has already created and repurposing them, we would need to understand this language. We would need to understand how to read and write it. And there's just a tremendous depth of complexity there. It's a language that's prior to all other languages and that ultimately distills down into basic biochemical and physical principles. So the methods that we're going to be using are these incredibly simple methods from a certain standpoint. And so one might wonder, how could this possibly work? And at this point, probably we're all familiar with what language modeling is, but just to remind all of you how simple a thing this is. One is given a sequence, and one trains a model to predict tokens in that sequence. And kind of the classic approach is autoregressive, so to be or not to be, that is the blank, and the model is asked to fill in the next token, given all the preceding tokens. Another variant of this that is also important is mass language modeling, where a number of different positions throughout the sequence are masked out. So to blank or not to blank, that is the blank, the model is asked to fill in those tokens. And so the training objective for these models is very, very simple. And what we've seen across artificial intelligence, and I will hope to convince you that this holds true in biology as well, is that as we predict tokens across very large data sets, which contain patterns that reflect sort of a deep structure, the models can discover this structure. And so that is the core idea, the core hypothesis behind ESM. And if I have time at the end, I'll talk a little bit more about why we might think this could work. But I want to begin by convincing you that it does work. And so if you think about evolution and sort of the vast scope of time and the immense set of experiments that are conducted by evolution in parallel and our ability to read those out through gene sequencing measurements, we have this view of biology, but only through the process of natural selection, which tokens evolution has chosen to place in sequences. And this idea dates back a very long time, that if we were to look at the patterns within protein sequences, and particularly within individual protein families, we would be able to see statistical correlations which would reflect the underlying biology. And this is shown here. It's an idea that's fundamental to AlphaFold, to methods of protein structure prediction that predated AlphaFold. And for example, if two positions in a protein sequence, which are far apart from each other in the sequence, but close in three-dimensional space so that they interact in some type of tertiary interface spatially, you can imagine that evolution is not free to choose their identity independently. And so because of this fact, if evolution switches the identity of one amino acid, it would have to make a change in the position that's in contact. And so we can see these patterns. And essentially, as soon as we were able to begin sequencing genes at a scale enough to look at multiple members of a protein family, these patterns became visible. And so, at a very basic level, one can imagine that it is this sort of thing that a model might begin to learn about. And so, we began looking at this, and we put out the first preprint describing ESM in the spring of 2019. And this is the core idea, which is basically we're going to train large transformer models across databases of millions, of hundreds of millions of protein sequences. And we'll ask these models to predict tokens across evolution, and then really look at what happens as the models learn to solve these tasks, and what representations, what properties develop and emerge within the representations of these models. And so one of the first things that you see that's, I think, very interesting is the emergence of protein structure in an interpretable form within the attention maps of these models. And so what is shown on this slide, on the left-hand side, the transformer proceeds through a mechanism that's called self-attention, where each position in the sequence is allowed to look at every other position, and sort of a weight is assigned to how much information will be transferred from one position to that central attending position. So you can imagine this as kind of synthesizing a graph along which information will be passed. And so as the input is processed through the model, block by block, in each of these blocks, self-attention will be performed, and for each input, a different graph will be synthesized. And so, the question is, what is the structure of this graph? And so you can look at the attention maps, and basically you will see that there are actually a set of attention heads, which across many different proteins, which differ greatly in their biology and their structure, there are particular attention heads which will correspond to contacts and long-range contacts in the three-dimensional structure of the protein. And this, of course, has just been learned from the sequences. So these models haven't been trained on structure. They've just been trained to predict amino acids. And one reason for this might be the logic that I described in the previous slide. But on the right-hand side, you can basically see a map of the attention heads in this model. So basically the bottom is the input layer of the model. The top is the output layer. Each of the heads are across the x-axis there. And so basically, there are the set of these attention heads that correspond to long-range contacts. And interesting, they're in the later layers of the network, so they're kind of synthesized later on. You can actually take this now and fit a really simple logistic regression, basically, to predict the contacts, and you get something that is essentially a state-of-the-art unsupervised contact predictor from this. So another thing that's really interesting is you can look at the probabilities that are output by the model. So you give an input sequence, and the model will give a probability for each token within that sequence. And so you can look at the uncertainty of or the entropy of the prediction for each token. And you can look at which tokens are predicted to be in each position. And we also see that there is a correspondence with biological function here. And again, you might consider this unsurprising in the sense that the tokens that are pure have been selected by evolution. Which tokens are feasible for evolution to place are tokens which are consistent with the function of the protein. But what we can see here, and this is a map of the model's entropy. And so if you just take and I believe these are the 10 lowest entropy positions in the model's prediction shown in blue here on a DNA methylase, these are clustering in the active site of the enzyme and in the residues that interact with DNA. So you can look at this more systematically. And that is shown here across a data set of deep mutational scanning experiments. And so the x-axis, different deep mutational scanning experiments. Some of these have hundreds to thousands to I think hundreds of thousands of mutations in them, depending on the data set. And here we're just looking at a log odds ratio. So sort of the log odds ratio of the mutated amino acid as estimated by the model versus the wild type as estimated by the model. And this is then a score for the effect of mutations, and you can just look at a Spearman correlation with the actual functional scores here. So this is, again, completely zero shot. We're not supervising the model with any information about function. We're just constructing a score based on the probability that it assigns to each amino acid in the sequence. The other thing that we saw, and this is from Lin et al. In this was pre-printed in the summer of 2022. We trained a series. So the first ESM model was a 700 million parameter model. And so at the time, that was a very large language model. It came out very recently after the BERT model and GPT-2, which were kind of at a similar level of scale. And so we're very interested in sort of what happens as you scale these models up. And one of the things that we saw even in the first ESM model is if you train models at different scales, you can actually track sort of a correspondence between the perplexity of the model, which is measuring its accuracy in its token prediction task, with the emergence of biological information, such as structural information. And you will see over the course of training, as the perplexity goes down, that the structural information increases, and also that as you scale models up, they can attain lower perplexities, and the structural information that they learn will increase. And so we saw that up to 750 million parameters. Here, we train models from 8 million parameters to 15 billion parameters, an entire family of models. And we looked for the signature of atomic level structure in these models. And so what I'm showing here is a very simple thing. It's basically you take the model after it's been trained, the representations are frozen, and now we're fitting an equivariant projection head onto this model. And that equivariant projection head is supervised with protein structure data, and it's outputting coordinates for each of the atoms in the protein. There are no folding blocks. There's no multiple sequence alignment. This is a very just simple projection. And so what we find is that there's enough information in these representations that you can project out, in many cases, a high resolution structure for the protein. And this information improves with scale. And this is just showing one example of this on a test set protein. One of the things that we see, I don't have the plot for this, but it's in the paper, is that at each jump between model scales, proteins will be learned at different scales, and so harder proteins will be learned later. And so you can see there's kind of a progression where this protein is learned, and actually it's learned relatively early here, primarily between the 35 million and 150 million parameter model. Okay. We've open sourced these models. One of the, I just want to highlight two, I think, really nice papers that we didn't have anything to do with, but that have explored ways that you can use these models. This is a study by Brandis et al. That looked at predicting the clinical effect of mutations. And so, taking those log odds scores and basically using them to score variants across the human genome, and they were able to find that ESM1B, and without using, again, any kind of multiple sequence alignment or anything, just kind of using those raw scores was a state-of-the-art predictor of mutational effect. This is a paper by Brian He and his team on optimizing antibodies with ESM. And again, this is really just using the token probabilities of the model. And it's a really exciting paper. They were able to improve the affinity of 7 different antibodies. They measured fewer than 20 new variants of each antibody across 2 rounds of evolution. So this is an extremely efficient process. And they were really just scoring these potential mutations using the probabilities that ESM outputs. These representations can enable very powerful predictive models. As we built ESM2 and we saw that there was an atomic level picture of protein structure in these models, the natural question is, can you use this to do single sequence structure prediction? Can you really kind of go from a raw sequence without multiple sequence alignment information directly to a 3D structure, and basically using a very simple head, using a series of blocks that have a similar kind of pairwise inductive bias to AlphaFold that use triangular attention, and some of those key mechanisms. You can put that on top of frozen ESM2 embeddings and get a state-of-the-art predictor of atomic level structure. And that's shown here on the Cameo test set. Accuracy approaches that of AlphaFold, but the inference is much faster here because we're not using multiple sequence alignments. We're just basically putting the sequence in and using ESM to produce embeddings and then project those embeddings into coordinates. There's also generalization that you can see where, for instance, the model can learn to predict interactions between different chains, even though it's not been trained explicitly to do that. So, one of the big questions I think that people have had, that the scientific community generally has had, I think that just naturally emerges from thinking about this predictive task is, can these models generalize? And it's a question that's been asked about language models in diverse domains, not just in biology, but more generally in vision, in language. It's very hard to ask this question rigorously in natural language because there's a sense in which the evaluation has an inherent subjectivity. But in biology, of course, we can query nature. We can look at this question empirically. And so, this is a picture of protein space. It's one which David Baker and Pasu Huang have proposed a picture like this to think about de novo protein design. And so you can imagine the space of natural proteins, which evolution has explored, occupying a very tiny fraction of the space of all possible proteins. And if we ran the experiment of evolution again, it may end up on, almost certainly would end up on a different set of proteins. And so, can these models really go beyond the set of natural proteins that nature has created for us already? And of course, if they couldn't, they would be, I think, much less interesting. So you can look at this question rigorously by asking, can these models create proteins that are distinct from, that really go beyond this space, that don't fall in existing natural protein families? And so we look at this in two different ways. The first way is we take a set of protein structures which have been created by de novo protein designers to differ from natural proteins in ways that they consider meaningful. So this could be different topologies or changes in the shape that are meaningful and that they consider to really be distinct from the structures that are used to realize those functions naturally by nature. So that's one test set that you can look at. The second way to really look at this is to just generally from the model and look at the sequence similarity to existing natural proteins and really ask, can the model find things that don't map to existing protein families in sequence space, but which do fold? So we look at this question in both of those ways. So the first slide here is showing the evaluation when we take a set of de novo structures that have been designed by protein designers. There's a test set here of 8 different structures. We expressed these proteins. We looked at 2 things. We looked at their solubility, and then we looked at size exclusion chromatography and the appearance of a peak at the expected molecular radius for the design. So you can look at both monodispersity or not, non-monodispersed proteins, which would which might be occupying more than one state. They might be dimers or trimers or so on. We're not explicitly designing for monomers, so we're considering it a success if we see a peak at the right molecular radius. And so we see a high success rate. And one thing to mention is that protein designers have seen this correlate with the existence of well-folded protein. So here we see a very high success rate just for designing for these backbones. And what we're doing is basically inverting the language model. So we're projecting out this low resolution picture of the 3D structure that I showed you before, and then we're optimizing the sequence so that contact map matches the contact map of the de novo protein backbone. And so we see an 80% success rate here for these designed backbones. So now this is looking at kind of the second way of doing this, which is generating sequences. And so here what we're optimizing, we're performing this kind of joint optimization where we're looking for sharpness, in a sense, in both the structure and the sequence probabilities. And so we're really looking for sort of a joint optimality between the sequence and structure. And so as we descend an energy well, so we're basically alternating, optimizing the structure and then optimizing the sequence. The structure optimization, again, is just in this projection space of the contact map You can actually see that in the animation on the bottom that just ran. And so we descend into an energy well. At the bottom of that energy well is a protein design. And so this is a map. The proteins are colored by their sequence similarity to the nearest natural protein. And so you can see that the distribution of these designs both overlaps with and is distinct from natural proteins, and that there are a diversity of different structures that are produced by the model. And again, if you put this to the experimental test, we saw a 55% overall success rate by size exclusion chromatography. And so finally, you can ask the question, okay, so out of that set, if you look at the set that is really distinct from natural proteins, and so that's shown in the lower left corner here, these are proteins which have a sequence identity less than 20% any natural protein for a number of them that are kind of at the 0 point on the x-axis. There's no significant similarity that can be detected. And then if you take each of them and you fold them, so you get a predicted structure and you search that against the natural protein database, now we're looking at the structural similarity of the nearest match on the y-axis. And so these are proteins which are distinct in sequence and structure. And here we also see a 63% success rate. So these, in a certain sense, these generations are no different from the generations which are much closer in sequence to natural proteins. I'm excited to present this. This is a crystallography result. So this is for one of the fixed backbone designs. And here we've been able to solve the structure for F003, which has a sequence identity of 22% to the nearest natural protein. Actually, I think this is to the template design. So because this is one of the fixed backbone designs, so this is a protein which de novo protein designers have created with a new topology. And so the model has found sort of a distinct solution from that which protein designers found, but which folds to this structure. So I think it's interesting to think about the implications of this, and maybe talk more about that, because there's some form of very interesting generalization that's happening here. I think you can't conclude from this, if the starting thought was that these models would memorize the existing protein families, I think that this contradicts that. Something more interesting must be happening. And what that could be is sort of an evolutionary grammar which links sequence to structure. There may be more than that, but I think at a minimum, you would have to conclude that there is something beyond sort of a memorization of the existing protein families. Okay, so I'm going to switch now, and in the remaining time, talk about ESM3. So ESM3 is our newest model. It's a model that's been trained now on another order of magnitude in scale. And so we train models from 1 billion parameters to close to 100 billion parameters on billions of protein sequences. ESM3 is a masked language model, but it differs from ESM1 and ESM2 in that it's a multimodal masked language model. So it's a model which reasons over sequence, structure, and function. And it's been trained with a generative masked language modeling objective. And so instead of being trained just to predict tokens that are masked out at 15% of the sequence, it's been trained to predict tokens across all possible mask rates. So because of this, you can generate a sequence using the chain rule of probability. So the model has been trained to predict the next token given an empty context, given 1 token, given 2 tokens, given 3, and so on, all the way to a sequence that only has 1 token missing. And so essentially, you can generate any token in any order and generate a sequence from that. Now, what's also interesting about this is it makes it promptable, because you can start the context with anything. And so if you're interested in conditioning the model on a certain structure or a certain functional motif, you can put that into the prompt, and you can ask the model to find completions that make sense. And so this has connections to discrete diffusion models. It's not a discrete diffusion model, but the chain rule factorization is something that ties those two approaches together. So kind of the key innovation that enabled this is the tokenization of protein structure. And so, basically being able to convert three-dimensional structure into discrete tokens that a language model can reason over. And so to do this, we created a VQ VAE autoencoder that basically maps local spatial neighborhoods in a protein into discrete tokens that compress their geometry. The decoder is then trained, given a sequence of tokens, to project that back into a full three-dimensional structure. This is enabled by a geometric attention mechanism, and this mechanism is essentially a variation on self-attention that is aware of the spatial relationships. And so attached to each position in the sequence is a reference frame. And so coordinates can be input locally to their reference frame, and in the attention mechanism, they're mapped into a global frame where they spatially interact. And so they can interact through dot products, so through similarity in their vector directions, and they can interact through distances so that close or far positions can communicate with each other. This can be very efficiently realized through the computational primitives of attention. So this is what the vocabulary looks like, and it's really kind of capturing these structural motifs that are local in space, and so you can see that it captures both local and tertiary motifs. The decoder delivers very strong reconstruction, and so for most of the test set, we see for the training of the decoder, we see RMSDs below 1 angstrom. So you can really compress the structure using these tokens. So as I said, we scaled these models up to 100 billion parameters. The largest model was trained on 1.8 trillion tokens overall, over 1 times 10 to the 24 flops of compute. So a tremendous amount of compute that went into supervising this generative model. And the data sets include metagenomic sequence data, the protein data bank, so real protein, empirically determined protein structures, and then a large amount of synthetic data as well, using predicted structures and inverse folded sequences. And you can think about this synthetic data. I think there's something very interesting about it, because in one of the directions, you go from synthetic to real. And so the model is supervised to predict a real sequence using synthetic prompts. And so I think there's some very interesting parallels to ideas in natural language processing around back translation, where you can use sort of a low resource language, and you can make a lot of predictions, and you can improve the ability to do translations. And so, using these types of principles, I think it's possible to, and this is a very rudimentary kind of form of programming to kind of generate these prompts, but you can imagine generating much more complex prompts synthetically. So I showed you the map of proteins with ESM2. This is the map with ESM3, unconditional generations. You can see there's a much higher level of complexity. The distribution both overlaps and is distinct from natural proteins in sequence space. The right-hand side shows histograms of similarity in sequence and similarity in structure. And what we can see is when we generate unconditionally, the generations are similar to the natural distribution and structure. They are distinct in sequence. When we, but what's interesting is when we begin prompting the model, we can steer it away from the natural distribution. And so here are shown two different types of prompting. One is for out of distribution folds, so these are folds that were not in the training set. The other is using these very idealized symmetric proteins, and no natural protein looks like them. They've been created by protein designers. And so you can push this model and it will generalize out of the natural space. I think the thing that's very exciting about this model is the promptability and the ability to compose prompts. And so here we looked at, just very systematically, can the model solve compositions of prompts that combine a high-level instruction about the fold of the protein with a very low-level description of a specific structural motif where we want to achieve an atomic accuracy in materializing that motif somewhere in the protein. So it's a scaffolding problem where we're saying, scaffold this motif, hold it in exactly this a particular atomic level configuration, and place that in a way that's consistent with a higher level instruction about the topology of this protein. And so this is an example of this. To make it concrete, you could prompt with an alpha beta hydrolase, and then ask the model to design specific set of atomic coordinates. So the model can do this systematically, and that is shown here, a variety of prompts and combinations of prompts. And in many cases here, so these are all good designs where the constrained RMSD, the atomic level RMSD between the prompt and the design is a good match. And we searched these motifs to see if we could find significant similarity to other proteins that contain the same motif, and for many of them, so the serotonin, calcium protease inhibitor, and MCL1 inhibitor binding sites, all of those, we couldn't find a natural protein that kind of had that same topology and that motif. The other thing that you can do with these models is you can align them. And so, I began by talking about the distribution of evolution. But when we're thinking about designing proteins, evolution hasn't, evolution is not thinking about proteins which are not natural and new functions. And so we can't, I think, expect the token probabilities to coincide with arbitrary functions that we might like to design. So in NLP more broadly, there's the idea of alignment, which is really how do you take capabilities that have been learned by the model that are kind of part of the distribution of the model in some way and elicit those capabilities in the model's generation. And so the way this works is you basically construct, it's learning by example. So you construct pairs of positive and negative examples, and you essentially supervise the model to put more likelihood on the positive examples while remaining close to the base model. And so using this, you can fine tune with a very small number of data points. So here we look at alignment to prompting fidelity, basically. So now we're looking at very challenging tertiary coordination tasks where we have to have parts of the sequence come together and interact spatially in very specific ways. And we're defining a success as having a PTM, so a strong prediction of the backbone above 0.8, and constrain RMSD in that coordination task of less than 1.5 angstroms. And the pass at K is basically measuring if you generate 128 examples of this task, or you generate 128 diverse generations for each task, what is the fraction that can be solved with 128 generations? So if one of them is good, then you'll get a solution at pass at K. So what you can see here is that there are differences between the models and their fidelity, the base models and their fidelity to the prompt. But when you apply this alignment, there's a latent capability that is elicited. And so the aligned models can solve double the atomic coordination task. And so we see this potential that's not revealed in the evaluation of the base model that can be elicited by basically a small number of examples that are used to teach the model. This also increases the diversity of the generations. That's shown here, the diversity before and after. And it shifts the overall distribution of the generations. The base model shown in red in these density plots, the aligned model shown in blue, upper left basically is the prompts that have both good CRMSD and high PTM. So I'm essentially out of time, so I think I'm going to have to just very quickly mention this final part, which is we really wanted to see can we use this to create new proteins. And so we started with GFP. I think GFP is interesting for two reasons. One, it's very easy to measure, so it's fast, it's easy to do, and two, it's a very beautiful mechanism that requires a very exquisite fine-tuning of function. And as far as we know, nature has only invented fluorescence one time. All fluorescent proteins kind of have this one mechanism, and it's an auto-catalyzed reaction. So the protein's backbone atoms reconfigure themselves into a chromophore. So it's very amazing. And so the prompt here is basically just the core coordinating residues, their atomic coordinates that basically realize this mechanism. And then we ask ESM3 to synthesize the rest of the protein. We use a chain of thought procedure, so basically we iterate between optimizing the sequence and the structure to find sort of jointly optimal sequences and structures. And so we did this with the 7 billion parameter model. Basically, as soon as we had that model, we were able to generate a plate of 96 designs. And these were bucketed, so we looked at things that were very close to AV GFP, the kind of the famous natural GFP that was the first to be discovered, and things that were far away. So you can see a lot of things that are lighting up kind of in the lower right there that are pretty similar to natural AV GFP. But what we're really intrigued by was this well B8, where there's a very dim fluorescent signal here. You had to be patient for it because it only appears on day 7, and it's 50x weaker than natural GFP. So you would barely see it. We weren't even sure, is this here? But there is some. You can see it as shown on the right in comparison to a chromophore knockout. And so we continue the chain of thought from that protein. So the key thing here is there's no kind of experimental data going in to fine-tune this model or to supervise a predictor or something here. We're just kind of using the structural principle, and we're asking the model to realize this structural motif. And so as we continue that chain of thought, and we made another plate of 96 designs, and I guess I should say about B8, why is B8 intriguing? Well, it has a 57% sequence identity. to the closest member of any, closest known natural fluorescent protein. So this is quite far away. And so we continued that chain of thought. And so on this next plate, we got a few bright hits, and the brightest is, well, C10 that's circled there. And this is 58% identity from the nearest fluorescent protein. So very distinct. It's really, the model has kind of jumped into an island in sequence space that doesn't exist in nature. So there's 96 mutations relative to the closest natural protein, or not, the closest known fluorescent protein, tag RFP. And the difference here is kind of similar to what you would see between proteins belonging to different orders within the same class. So like the difference between stony corals and sea anemones within Anthozoa. So if you kind of look at this and ask the question, sort of a thought experiment, like if evolution were to generate this, what would it take? And you look at this divergence has occurred over predictable timescales in evolution. This would essentially equate to something like 500 million years of evolution. So I'll conclude there. I won't have any remarks on, why this can be, but I would like to acknowledge, the tremendous amount of work and collaboration that went into this, especially the work on generalizing beyond natural proteins, which is in collaboration with Sergei Ovchinnikov and a team at the Institute for Protein Design, and then ESM3 was created by this team at Evolutionary Scale.
Thank you so much, Alex, for a really, really wonderful talk showing us how far you can push it by such clean models, and really models where you're thinking about very carefully and seeing what you can just get out from there and all the structures. So we have time for questions. And we'll have people moving around again. And I don't really see, but. So maybe, well, do you want to start? Yeah, okay.
Thank you, great talk. It's amazing to see the power of a language model for protein structure prediction. So from perspective of first principle, it is equivalent to solve a multi-body Schrodinger equation. So can we conversely think that we can use language model of protein prediction as to decompose this model as a simulation of quantum computation?
It's a really interesting question, and it's what I would have talked about in my slides if I hadn't continued. So I'll say a few things about this. I think it's a very different kind of simulator that we're creating here. So the classic idea of a simulator is really to write down the world in basic principles and equations and follow their evolution to understand what are the possible future states. This is very different. It's an information theoretic model, I guess. It's a predictive model, and so it's really looking at the correspondence between input and output. There's maybe some sense in which, in the limit of infinite data, these two things coincide. I think the thing that I have found to be very there is an idea, I think, that has been behind the development of all of this, which is basically, if you consider the next token, it's generated by evolution. And how does evolution select the next token? The entire deep complexity of biology is behind that decision, because it selects tokens which do not lead to the collective loss of function of the system in which that protein participates. So to solve that task well, you would imagine that if the model can generalize, if it can predict the next token with fidelity, it would have had to develop representations that are capturing and modeling some of that deep biology. So that has been the core hypothesis, that information would exist within the representations of the models. And I think it's very important to think it's not in the token probability. The token probability is the probability under evolution. But to solve that task, it would have to develop a feature space which captures the depth of biology. Now, whether it can go that far, I don't know, and so I don't want to say anything about that. That's an empirical question. Perhaps with enough data, it would be possible. But I think there is a principle here where there's a distributional hypothesis of biology. The data is generated by evolution. There's a deep structure to it, and the model to solve that task would learn something of the deep structure.
Thank you.
Can I follow up on one of these comments, because it was also brought up before in talks? So in terms of like the synthetic data that you have used, it still seems very small as compared to the full data that you were using for training. So first of all, were you worried about using more? Why maybe not using more? And then also the other question on, that's on the data side and on the, scaling on the data side, but then also on the scaling on the parameter side, which you've done a lot of. What do you see in terms of laws that are emerging, and where are you in terms of like, do you need to build much larger models, or do you see any tapering off as well? Yeah,
okay, so on the question of data, well, we always want more data, I think, is the short answer to that question. So it's never enough. And we want the data to be better. So I think the other reason for these models, of course, is that there is so much sequence data, and that's growing exponentially. We can expect it to continue to grow. We can expect it to encompass so much more, like cell states, and we've heard about those models. So there's this tremendous growth of data. We're not seeing that same growth in protein structure data, so there's kind of a limit there. So I think predictive models are really exciting because they've kind of expanded our ability to understand structure, to train these models. I think there is some issue which you would expect to see some saturation because, in a certain sense, the model could learn to become a simulator of AlphaFold with enough data lake instead of becoming, a simulator of the actual structures. So I think there's a lot of deep research to kind of figure out how do we use data augmentation effectively here. But yeah, of course, we would want eventually to fold all the proteins in the training set and have sort of sequence and structure and functional data for everything. And then on saturation, we don't see it. And I think, and I think in principle, like we train this model on 2.78 billion protein sequences, I think. And so there are many more. So it's, I think that, there are many, many more. I mean, how you select them matters. Having good tokens matters. So I think, I think it's an interesting question how far we can go. But I guess I would say I think we can go further with the data that we know is there.
Thank you for this really interesting talk. I was wondering, how does your model account for variations in sequence length? Like say when you're trying to predict the next token, does it have to be one amino acid, or can it be a deletion, or can it be multiple amino acids?
So in principle, you could do that. But yeah, the way this is set up is you would have to say how long the protein is because it's not predicting an EOS token. But you could have it predict an EOS token if you wanted it to do that. But ESM3 hasn't been trained that way. So you put in a certain context length when you're generating.
What do you need a lot more training data to account for variations in sequence length in the model?
No, I don't think at all. I mean, the model takes, of course, sequences of all different lengths because the transformer can, would process them all. It's not a fixed context length. So,
okay, thank you.
Okay, so thank you, Alex, for a really, really wonderful talk. I hope you'll be here for lunch to take on some more questions.
Absolutely.
And with this, let's thank Alex one more time. And we'll all be moving out for lunch and meeting back here again at 1:15 p.m.